You could start to slow down. So you could probably start whenever you'd like.
Yeah.
Okay.
So, welcome everyone to today's Club EVMed. My name is Henry Schulenburg from the University of Kiel, and I'm chairing today's session. Yeah, just as a reminder, the goal of Club EVMed is to connect evolutionary medicine scientists across the world and yeah, share our exciting research. And we try to be as informal as possible and with an emphasis on discussion. So you're very welcome. I'd like to invite you to actually join the discussion after the presentation. Also, as a reminder, the Club EVMed is organized by the International Society for Evolution Medicine and Public Health. And it would be great if you generally would support the society by becoming a member and or by joining the society's annual meeting, which will take place this year in Durham in the UK from 6th to 9th of August. I should add that Club EVMed is additionally supported and organized by more than 10 other evolutionary medicine centers and programs across the world. So we will now first have a talk, and then there is an opportunity to ask questions to Csaba Pal. And you can raise your hand or post the questions into the chat. And I tried, and I give my best so that everybody can actually ask their question and get a reply from our speaker today. This is, I think, all for the introduction. Now it is my great pleasure to introduce Csaba Pal to you. Csaba did his doctorate in 2018. This cannot be true. Sometime earlier.
Slightly earlier, but thank you.
Yeah, I don't know where I got
I'm
that
not
number
that
from.
young.
Okay, you did your doctorate sometime
Yeah, that's
at the
good.
Hungarian Academy of Sciences in Budapest. And then you continued as a postdoctoral fellow funded by the Royal Society in the UK, where you then joined the group of Lawrence Hurst at the University of Bath. You then did another postdoctoral fellowship at the EMBL in Germany, followed by an EMBO postdoc fellowship at University of Oxford back in the UK. And you've been a visiting scientist at the University of Trento. In 2008, so currently 10 years before you did your doctorate, you already started your lab as a PI on synthetic and systems biology at the Institute of Biochemistry in Szeged, which I may mispronounce, so you have to correct that in a minute.
That's
and
okay.
Since then, you have established a research program, a very ambitious and competitive research program, with one focus being antibiotic resistance evolution research, but also covering a couple of other topics. In 2020, you became head of the antibiotic research program of the National Laboratory of Biotechnology in Hungary. Csaba has been awarded a couple of quite prestigious prizes, including the Ignaz Lieben Prize by the Austrian Academy of Sciences in 2008, the Gent-György Talentum Prize in 2014, and the Volyai Prize in 2015. And recently you have been elected an EMBO member. It's also very impressive that Csaba got both an ERC starting grant and a consolidator grant. And most recently, this was only announced last week, Csaba actually was awarded the highly prestigious ERC Advanced Grant by the European Research Council. So congratulations for that, that's still very fresh. And what is super exciting is that you got that ERC Advanced Grant on the topic directly related to today's talk. So now the floor is yours. We hear about antibiotics of the future, and I'm very much looking forward to your presentation.
Thank you for these kind words. I'm really honored to be here and very much looking forward to your questions and concerns and criticism. And this is indeed pretty much what I'm planning to do in the near future. And what I'm trying to show you is some of the preliminary results, what we have on this topic. And as a matter of fact, this is a topic that it's very easy to communicate because it affects all of us. And I think it's, something that it may seem very trivial, that there are many new antibiotics which will reach the market pretty soon, or some of them are at a preclinical stage. And the question is whether they will be effective in the long term. And if yes, why? If not, why? What are the major mechanisms that affect the evolution of resistance against antibiotics, and how can we design better antibiotics? So this is pretty much the goal. So with that, I would get to my first slide, that, this is a somewhat paradoxical situation we are facing with at the moment, because on one hand, all of us are aware that antibiotic resistance is a huge problem and a global health concern. But on the other hand, the several big pharmas have abandoned their antibiotic development program for quite a few reasons, but one of them is due to the rapid emergence of resistance in patients during clinical tests. So many of these companies have actually spent hundred millions of dollars into a single antibiotic candidate, which turned out, and they had to abandon their programs because in late clinical phases, phase 2, it turned out that these antibiotic are extremely prone to resistance. Of course, there are other issues, but this is one reason why many pharmas consider that developing new antibiotics is a very risky business or endeavor. On the other hand, if you read academic journals and some of the most prestigious journals like Nature, Science, etc., there are many works which claim the discovery of resistance-free antibiotics. But if you actually consider the data underlying these claims, you'll find that the data is actually very limited. And both issues stem from the same problem, because if you consider the standard laboratory technologies to forecast resistant evolution, then you will find that they have certain limitations, including low throughput, which affect the number of pathogens and the antibiotic candidates that can be tested. And as a result, there's a limited exploration of the sequence space, and most of the technologies ignore horizontal gene transfer altogether and focus on resistance by genomic mutations only. And of course, even if resistance found in the laboratory, it's unknown whether these resistance mechanisms have any clinical significance. So this is somewhat, it's a very important issue to overcome these problems. And what we actually believe is that we have quite a few new methodologies that could contribute to better predict evolution of resistance against new antibiotic candidates. So this is basically the major issue that we are interested in. For this purpose, we generally study new antibiotic candidates which have recently reached the market or at a clinical phase 1 or 2, or they are preclinical stage. And we specifically focus on molecules and compounds which are claimed to have new molecular targets, new chemical structure, or special mode of entry into the cell, and they were actually claimed to bypass existing resistance mechanisms. And we also study other antibiotics as a control, which have been on the market for a number of years. And very simply, we wanted to test whether resistance emerged in the laboratory against these new antibiotics as rapidly as against clinically employed antibiotics. So in our laboratory, we use quite a few different technologies. And one of them is rather trivial, laboratory evolution, where you basically increase the dosage of the antibiotics on a daily basis, and the bacteria population in the test tube must adapt by new mutations, and these mutant variants will emerge during the course of laboratory evolution, enabling the population to survive higher and higher dosages. In addition, we use state-of-the-art genomic engineering technologies, which will allow us to study directed evolution at specific loci. So basically generating very high mutant variant, very high level of mutation rate at specific genomic loci only. This is applicable only if you know which are the interesting loci that, which are the loci which might be involved in resistance. And of course, we aim to study resistance by horizontal gene transfer. And for this purpose, we use the specific protocol, what we call DeepMind. This is a new version of functional metagenomics that allows you to explore mobile resistant genes in the environment, which actually can function and provide resistance to new bacterial hosts, including pathogenic hosts. So today I will generally focus on the simplest, on some of the results what we have generated by laboratory evolution. And in total, we have roughly 400 laboratory evolved bacterial lines. And we, of course, sequence the lines to identify the de novo resistant mutations. We study 4 bacterial pathogens with critical clinical importance, including Acinetobacter baumannii, pathogenic version of E. coli, Klebsiella, and Pseudomonas. So what you see on this graph is actually striking. On the x-axis, you see the MIC of the ancestor, the minimum inhibitory of the ancestor bacteria. And after roughly 100 generations, we generated laboratory evolved bacteria. And here on the y-axis, you find the MIC of the mutant lines, the laboratory evolved lines. And basically for nearly all antibiotics we considered, we found a massive increase in the minimum inhibitory concentration. In general, at least a 64-fold change in resistance level, sometimes by over a resistance level which was above the clinical break point. Actually, not just sometimes, in most of the cases, we find that the resistance level reached was above the clinical break point. And on average, we found that new and clinically employed antibiotics are equally prone to bacterial resistance. So from this point of view, it turns out that new antibiotics are as prone to resistance as the old antibiotics are. So this is a pretty bad news. Just let me give you just one example. One of them is teixobactin, which is actually an antibiotic developed against gram-positive pathogens like Staphylococcus aureus. And in a seminal paper, Kim Lewis and colleagues claim that this is a new antibiotic that kills pathogens without detectable resistance. And rather, unfortunately, we found high level of resistance against teixobactin using our methodologies. So it appears that many of these antibiotics actually are prone to resistance. So when we sequence the evolved lines, we generally find roughly 5 to 10 mutations, putative resistance mutations that provide resistance to the laboratory evolved bacteria. And when we compare these mutations or the mutant genes, we actually find a substantial overlap in the molecular mechanisms of resistance between clinically employed antibiotics and new antibiotic candidates. This is actually, again, a bad news because it suggests that if there's an overlap in resistance mechanisms, it actually predicts that the observed mutations providing resistance to new antibiotics may be already present in the environment or in clinical settings, even though these antibiotics have not reached the market yet. So we were very much interested in whether the laboratory observed mutations against these new antibiotic candidates can already be found in clinical isolates or in natural bacteria. So for this purpose, what we did was that we used a catalog of bacterial genomes derived from natural isolates of E. coli and Acinetobacter baumannii. And using standard protocols of bioinformatics, we actually found that over 30% of the resistance mutations found in the laboratory are actually already in natural or clinical isolates. So this actually suggests that in many cases, there are resistant bacteria to these new antibiotics, and pre-existent these mutations pre-exist, and they can spread rapidly once these antibiotics reach the market. However, there are some good news as well. One of the good news is that we found a very strong tendency for species-specific resistance, which actually means that high level of resistance emerges in specific bacterial species or strains only, while in the others, we fail to find any high level of resistance. So it suggests that there's an immense differences in the capacity to develop resistance against certain new antibiotic candidates. Why it may be the case? Well, we don't know yet, but there could be at least 3 different hypotheses. One of them is there are major differences in the initial antibiotic susceptibility across bacterial species and strains, and therefore these initial differences in antibiotic susceptibility may affect later evolution of resistance. Which actually I doubt. Our preliminary data suggests that this is not the full answer. But we also find that there could be some species-specific resistant genes and mutations which affect further evolution of resistance. So there could be actually some sort of epistasis between pre-existing mutations, resistance mutations, and the ones that emerge later. And finally, and this is somewhat related to the fact, some certain mutations and genes affect mutational effects and whether they will provide resistance. Okay, the other good news is that we actually found certain antibiotic candidates with limited resistance. And apparently, even though they are structurally and chemically, they are very different from each other, they've got some common properties in common. They actually fulfill two criteria simultaneously. So they are dual target antibiotics. They've got they've got 2 main molecular targets within the cell. And secondly, but that was not enough. The other common property of these is that they actually disrupted the bacterial membrane simultaneously. So this is the group what we call dual target membrane disruptors. And for these dual target membrane disruptors, we found, as I said, we found limited evolution of resistance. And we are keen to understand why they are so, what makes them free from resistance. One possibility is that resistance mutations and mobile resistant genes may be rare against these antibiotics, or these mutations may emerge readily, but such mutations would have a very high fitness cost. So it would affect the carrying organism, either the viability or the virulence, and therefore they cannot spread in nature or in the test tube. So this is something that actually brings us to the second question, which I'm particularly interested in: how resistance to new antibiotics shape bacterial virulence. Now, there are quite a few scenarios which you can consider. The optimistic scenario is that as resistance emerges, it has a fitness cost, and there will be a negative trade-off between resistance level and virulence. If it were the case, then it would be, one might argue that with high level of resistance, many of these candidates would not be viable, and therefore these antibiotics could be effective in the long term, even though resistance could emerge, but it's clinically irrelevant. However, there's another possibility, is that this is what we call a pessimistic scenario. That antibiotic, sorry, yeah, okay, that there is a positive trade-off. So when the very mutations that provide resistance at the same time will actually increases virulence. And of course, we don't know yet whether this is the case, but in our laboratory, we implemented at least 3 different technologies to study virulence. So in one hand, we've got a very interesting invertebrate infection model. We also study bacterial invasion of human cell lines by bacteria, by basically co-culturing human epithelial cells and bacterial pathogens. And of course, once we have preliminary results with these first 2 technologies, we actually test it further with mice infection models. And I really don't want to get into the details for quite a few reasons, because this is actually preliminary. I would just like to give you one example. This is SPR206. SPR206 is a new antibiotic candidate. This is peptide-based antibiotics currently in clinical phase 2, developed by Sparrow Therapeutics. And it has an excellent antibacterial activity against gram-negative pathogens. But rather unfortunately, we found that resistance rapidly evolves against these antibiotic candidates in the laboratory. And there are quite a few resistant genes which are present in a range of human-associated microbiomes. Both of these data are coming from our laboratory. Now, even worse, when we consider these laboratory-evolved bacteria that are resistant to SPR206, we found that they displayed cross-resistance to colistin, another peptide antibiotic, which is actually a last resort antibiotics. And these SPR206 resistant mutations provided enhanced bacterial virulence in multiple infection models, including mice. So again, this is still somewhat preliminary, but this is we find that it's potentially an important concern, because it suggests that once these antibiotics reaches the market, it may generate hypervirulent and multi-drug resistant bacteria. So clearly, we need to see whether such preliminary results hold more generally and for other antibiotic candidates, or is it something that is specific to the certain peptide-based antibiotics only? So in sum, our data suggests that antibiotic candidates which are currently under development are actually very prone to resistance evolution in gram-negative pathogens. And these resistance mechanisms are actually already present in certain natural populations of bacterial population, indicating that resistance can emerge through selection of pre-existing bacterial variants. And as I said, there's a better news that there are certain antibiotics that for which resistance appears to be more limited, and this is probably a good direction for future antibiotic development endeavors. And last but not least, the capacity to develop resistance is species-specific, which actually emphasizes the potential of narrow-spectrum antibacterial therapies that may remain effective. And of course, the big question is whether whether antibiotic resistance mutations may promote enhanced bacterial virulence, and if it's so, whether it's a general phenomenon or not. With this, I would like to thank for all my colleagues who has been working on these projects for quite a number of years, and I've got excellent collaborators as well. And of course, I'm very happy with the fundings I have at the moment. Thank you for your attention, and I'm very much looking forward to your questions.
Thanks a lot, Csaba. This was a very exciting talk. So the floor is open for questions, and I suggest that we start with the first question that was pasted in, put into the chat. Neil Greenspan, do you want to ask the question yourself?
Sure. Can you hear me?
Yes, sure.
Yeah. So I asked if any of the mutation, any of the resistance you identified might be due to non-mutational, standard mutational mechanisms, because there have been enough. In fact, I think it's kind of confusing now as to what is tolerance versus permissivity, or whatever the other word was for more, non-mutational mechanisms. I don't think that undermines your point at all, but I just think it might be interesting to know what range of mechanisms bacteria might employ to minimize the toxicity of these antibiotics.
Mm-hmm. Well, shortly, I don't know the answer yet. This is still, an investigation ongoing. So it's
But I mean,
clearly.
do you have any interest in looking for these other kinds of changes in bacteria that mediate some level of resistance?
What other mechanisms you have exactly in your mind, if I may ask?
Mechanisms that involve relative metabolic quiescence, those sorts of.
Oh yeah. So there's one interesting that we find. For a range of antibiotics, we find mutations which are known to affect the stringent response. And this was something, and the stringent response has been known for some time to have an effect on antibiotic tolerance for certain antibiotics. But actually, our data suggests that this is far more general, and it's actually could be the first step or has an important role for developing resistance to a range of new antibiotics as well. So this is something that we are investigating at the moment to identify how exactly these mutations, stringent response genes, affect resistance, and what are the side effects of them. So this is another side project that we are investigating at the moment.
One other comment I'll just quickly make is that I think your interest in assessing the effects of mutations that confer resistance on virulence is extremely important and interesting. But wouldn't it be reasonable to suggest that there are many different dimensions of virulence, and you could have differing effects of mutation that confer antibiotic resistance that would increase one element and decrease another? So it could get very complex to sort out.
Yeah, I absolutely agree with you. So somehow we have to focus on what aspect of virulence we are focusing on. I can certainly imagine, for example, that certain resistance mutations will disrupt, let's say, biofilm formation, but increase, let's say, colonization of human cell lines. And actually, therefore, we actually try to investigate quite a few different aspects of virulence. And I'm very much looking forward to see whether there are actually negative and negative trade-offs between different aspects of virulence as well. But at the end of the day, I think we need to go to, a real world situation and to ask how it will affect infectivity and how it will affect the capacity of these bacteria to cause disease in an animal infection model. But yes, I'm very much interested in this issue, and I fully agree I oversimplified this question. Yes.
Thank you.
I think related to this topic was the comment in the chat by Melody Jiao. I don't know whether you would like to quickly raise that point.
So the question was, I think I will read it to the audience.
Yeah.
The Davis lab at Hopkins does similar work with pseudotuberculosis, possible a good model to test these next generation antibiotics. Yeah, I think absolutely. I'm generally fascinated by including more and more different bacterial pathogens in the laboratory. In general, because I think that the more you include, the more nuanced answer you will find. So, it was really fun to see that certain bacterial species show such a high level of resistance developing in the laboratory, not the initial level of resistance, but the evolution was very rapid for certain bacterial species only. So clearly, I have to read more about this work and see whether this would be a good model. So I think I gave only a very general answer, not specific to this bug. Okay.
Then Lotus as well did ask a question. Do you want to?
So maybe I should go further. Okay.
No, yeah, I would try to engage the people who raised the question. So if they want to ask them themselves,
Okay.
Okay.
So
you're welcome. And Lodi as well.
Thank you. Yeah, I was wondering if those antibiotics, the SPR206, was already used either in the field or in the lab on other mammals like livestock and pets. And I can make the link to the other question, which is related to AMR, obviously. So yeah.
So it has not reached the market yet, and as far as I know, it's still in the experimental stage. And I'm not aware of that yet. So yeah. So what is very important, I don't want to criticize the company or the molecule per se. I'm just trying to highlight a general issue because this may be more general. And what I actually trying to say with this specific example is that you have to be really, really careful when you work with these antibiotics, and you have to test resistance emergence and the side effect of resistance at a very early stage of development. Otherwise, you might find yourself in an awkward situation that you spend hundreds of millions of dollars as a company leader into a given molecule, which may turn out that will be actually dangerous to use in the long term. So this is what I'm actually trying to say with this. And I don't want to claim this is actually the case, because I try to highlight that we are still at an early stage with SPR206, but our preliminary data clearly indicates that this can actually happen.
Okay, another question comes from Indranil Samantha. Indranil, do you want to?
Yeah, is there any possibility of linkage between antimicrobial resistant gene and virulence gene, maybe present in the same mobile genetic elements? It actually happens very often. This is a well-known fact that on the same plasmid you find virulence genes and antimicrobial resistant genes. Whether it holds for our antibiotics, I don't know yet. But this is what I'm actually suggesting is somewhat different. What I'm actually trying to investigate is not necessarily genetic linkage, but a given gene or a given mutation provides resistance and virulence at the same time. And I did not have time to get into that, but we actually identified quite a few mechanistic links between the two phenomena. And this was especially so with peptide antibiotics, where resistance emerges by actually changing the bacterial membrane potential or other aspects of the bacterial membranes, which also have a side effect on bacterial virulence.
Okay, thanks. I'm still trying, Csaba, that the person who posted the question can ask the question themselves to get them more engaged. So with the next person we try, James Gurney.
Hi, how's it going?
Hi, James.
Hi, how you doing? So I just got stuck on this idea that the resistance evolution is species specific as opposed to, say, the standing variation. I still don't understand why, say, E. coli or might have a lower ability to evolve resistance than, say, Pseudomonas outside of pre-existing resistance strategies that Pseudomonas might have.
Yeah, I think it's our data indicates that it's not due to pre-existing variation, and it's not due to intrinsic differences in the susceptibility, let's say between Pseudomonas or E. coli. One trivial answer would be that differences in genomic mutation rate, But this is again was not the case, because for actually we measured it and we know that this was not.
What about
yeah?
the things such as the ability to excrete or change the environment in terms of how much of the genome is dedicated to, Pseudomonas has an awfully large genome that's dedicated to cooperative behaviors that are typically excreted outside the cell. And that's also linked to the idea that it's more of a generalist than, say, some other pathogens. Did you look at a correlation in terms of like how much of a generalist a pathogen is compared to, say, a more specialist pathogen?
Mm-hmm. Well, I don't know yet, because for simple reason, when you use laboratory evolution, this is a very simple environment compared to realistic clinical settings, and I readily admit. The reason why I believe it still has something to say about what happens in nature is because of this high overlap between the observed mutations in the clinics and in the laboratory. But getting back to your question, we actually think we have some hypotheses. What we can actually try to identify is what we call potentiator genes. Potentiator genes have a very limited effect on resistance per se, but they affect the impact of new mutations. So, for example, efflux pumps. There are certain efflux pumps which are species specific, and we know it very well in case of Pseudomonas. And the presence or absence of these efflux pumps will have a very limited impact on, or relatively little effect on, resistance per se. But if a target mutations occurs, the two together, the efflux pump mutation and the target mutation, would drastically increase resistance level. And this is something that is known for quite a few antibiotics which have been employed in the clinics. And what we believe that this is what happens. The presence of these genetic changes in one or other strains or species will affect the impact of new mutations. So this is what I'm actually thinking about at the moment. Well, if you think it's nonsense, I mean, I'm happy to take your criticism.
No, but we related to that because I also found it very interesting that you seem to have some data that resistance evolution, or the potential to evolve resistance, is species specific. At the same time, when we look at Pseudomonas, for example, then most of the work is done with 2 laboratory strains that are known to actually have adapted to laboratory conditions, so PA1 and PA14.
Yeah.
So did you use those? And the question is, perhaps you would get a very different result if you used sort of more natural isolates of the species.
Okay, so I think this is an important general concern. What are these mutations doing? Whether these mutations are actually, what was the adaptation? Was it adaptation to the physical environment? Because if it was to the nutrient conditions, then most of the mutations would be irrelevant. And yeah, one of the reviewers raised this issue, and I think it was very important. So what we did, we designed new experiments, which were the same experimental settings without any antibiotics added. So we did exactly everything was the same, just no antibiotics. We actually sequenced the evolved lines, consider the mutations, and compare the mutational set in these control experiments versus in the antibiotic evolved bacteria, and there was practically no overlap. So what it actually suggests that in our experiment, it's not about adaptation to the nutrient condition or to the physical environment. It was really about the new antibiotics per se. But yeah, it's a very important issue. I fully agree.
Yeah, I think, so Neil, I saw your hand. I just have my add-on question. I was perhaps not thinking about specific or the adaptation to the physical environment, but rather the genome content, because it is well known for Pseudomonas aeruginosa that the species has an enormous pangenome. and you find enormous differences between different strains. So it could well be that if you use a different genomic background, there may be a completely different potential
Yes.
to adapt to these antibiotics.
Absolutely. So what I think, this is you can have two experimental strategies. Consider large number of antibiotics and relatively few strains. So what we actually use, we use 2 strains per species. Altogether, 8 different, 4 species, 2 strains per species. The other strategy could be, and this is what we are trying to do, is to consider only 1 species, but at least 40, 50 representative strains of the same species evolving to a single antibiotics and see whether there are indeed major differences in the capacity to develop resistance.
Okay. Cool.
And this would give us a better answer to one antibiotic.
Yeah.
Good point. We took the other took the other approach. We were interested in to get a general answer to a range of antibiotics instead of focusing on a range of bacterial pathogens.
Okay, cool. Neil.
Yeah, so I had another question about whether you would have an interest in looking, and maybe it's hard for any one lab to do, looking at the potential trade-offs between antibiotic resistance and susceptibility to either immune mechanisms like complement or phage. Because, what I've found going back 25 years, I had done some research on potential problem with Pseudomonas in the context of cystic fibrosis many years ago. And our approach to address a particular problem that had been described at that point, this is back in the 90s, was to create a bispecific antibody that linked a complement split product to the right complement receptor, because neutrophil elastase destroyed the receptor in one case of the pair and the ligand in the other case. Now, that's just one possible approach. There are many other approaches, but the people who work on immune mechanisms never deal with antibiotic resistance, and the people who work on antibiotic resistance never seem to deal with the immune aspects. And then there's the whole phage therapy.
I fully agree. Yeah, that's really odd, and I think it's a brilliant question and suggestion. So this is something that I should do in the future. I have not done it yet, I fully admit. As regards phages, we actually have a work where we are interested in the trade-off between antibiotics resistance and phage susceptibility. So we've got a range of natural phages from different environments, and we do see negative trade-offs. So there's a collateral sensitivity between, at least in certain cases, between phage resistance and antibiotic resistance, which would suggest that the combination therapy may work.
Right. Thank you very much.
But as regards the complement system, my knowledge is very limited. What I have done is that, and this is something that I'm particularly interested in, to study natural antimicrobial peptides, which are found in the human body, like defensins, and yes, we measured in vitro the susceptibility. And in general, especially with peptide-based antibiotics, we unfortunately find a positive link. So basically, they are also the SPR206 and other peptide antibiotic-resistant bacteria also show resistance to human peptides involved in innate immune defense.
Thank you.
As I see, I'm getting quite a few questions. I'm really trying to answer all of them. And I'm really grateful for your questions, guys.
I think we have a little bit of more time for this. So Cynthia Beale rather had a comment. Do you want to, Cynthia?
Sure. I'm following up on, Henrik, your comment about bacteria adapting to laboratory conditions made me wonder if an interesting angle would be to take a half dozen clinical isolates from a half dozen patients who would have different be providing different environments for the pathogens.
Absolutely. Absolutely. As I said, the strategy would be very different. Consider a wide range of clinical isolates from different patients, possibly different conditions, from the same species, and then let's do the laboratory evolution whether to see how fast they can evolve and whether there are any differences, and if so, what the mechanism might be for these differences. Yeah, absolutely. So quite honestly, the result that we see any differences in species-specific differences came as a side effect of our project. At first, I just wanted to demonstrate that what we have is a general effect when we see resistance level in basically all species or no species. And to our biggest surprise, we found huge differences across bacterial species and strains. So there are certain strain and antibiotic combinations with limited resistance, but there were practically no antibiotics for which no resistance emerged. So there were no resistance-free antibiotics in our data set. Yeah, but yeah.
Thanks.
So in my opinion, another interesting aspect could be if you get resistance against sort of more challenging antibiotics, like the dual target membrane disruptor antibiotics, is that they may cause quite high costs to the resistant cells, which as a consequence could mean that resistance is also rapidly lost if you would not take these the resistant bacteria and evolve them in the absence of antibiotics.
Yeah.
Did you assess something like that? So the general growth cost of resistance and how stable the resistance is.
Yeah. So actually we we did it systematically for hundreds of strains. First, we measured the in vitro fitness cost. So basically just standard laboratory conditions, see how fast they can multiply and grow using standard measured of fitness. And yes, in basically the result that emerged that in roughly half of the cases, there were no costs whatsoever, and still we find some strains where we see a positive, may I call it a positive trade-off? I don't know what's the best word. So basically, they were growing rapidly than the original wild type. And we also studied the same phenomenon in Galeria. The good thing, which is a invertebrate infection model. And the good thing about Galeria is that you can use it high throughput. So you can study hundreds of strains simultaneously and ask how well they can actually infect Galeria, where the output is basically the survival of the larvae. And basically, again, 50% of the laboratory evolved lines displayed enhanced infectivity and higher killing rate and these are the cases, and of course we also see negative trade-offs in quite a few cases. So clearly we want to differentiate why and for which antibiotics we see a negative trade-offs and how often we find that virulence actually increases or decreases.
Just
and
out
how
of
it depends
curiosity,
out
of curiosity, the resistance you found against the new substance SP something, where you mentioned that it also shows cross resistance to colistin, and at least some of our own data with Pseudomonas indicates that resistance to colistin is not so stable. Was that a case where you also found instability of the resistance, or meaning high cost of resistance?
Well, I don't know what stability really means. It was stable for a few generations, and we could sequence it. We see that it remains for a couple of generations. For long-term stability, it's actually what you need to know is that you need to grow them without adding any antibiotics for hundreds of generations, and see whether resistance will remain intact or will actually disappear in the long term because of the cost. And we did it with another project which was published in eLife, I don't know, 3 or 4 years ago. And for quite a few antibiotics, we see that resistance actually remains but the cost is diminished. Of course,
Okay.
compensatory mutations will emerge in these populations.
Okay, thank you. Are there any more questions? I think in the chat there have been none. If that is not the case, then I thank everyone for joining in today, and big thanks go to Csaba. That was a pleasure to hear about your work and also to join in with you for the discussion.
Okay, thank you very much. It was a pleasure being here with you. There's actually a new message. Thank you. Okay, thank you for the comment. It's very kind of you, all of you, and I was really happy. I'm very enthusiastic about this work. So if you have any other further comments or you think that there would be a point for collaboration, I would be really honored. And yeah, so thank you for all the feedback. That was really kind of you guys. Thank you.
I think last but not least, before all of you log off, I'd like to announce that the next talk of the EVMAT series will be on the 2nd of May, Thursday, 2nd of May, same time, same place. And it'll be another one, another talk in the series on antibiotic resistance evolution, and this time by Pamela Yee, and I actually apologize, I have to look up, and Dr. Sada Boyd. From UCLA in the US, and again on some aspect of antibiotic resistance evolution. Thanks again, and see you all soon.
Yeah, thank you. Bye now.